Welcome to our channel, where history and science meet to uncover the hidden wonders shaping our future. In today's journey, we explore the genetic blueprint of the remarkable Moringa tree. Discover how this ancient plant's DNA could transform modern medicine and revolutionize healthcare worldwide. Stay tuned for captivating insights. Can Moringa's DNA revolutionize medicine? Unlocking nature's genetic blueprint for healing. Part 1. The ancient roots of Moringa. From the dry, sun-scorched landscapes of the Indian subcontinent emerged a tree that would one day draw the gaze of healers, botanists, and geneticists alike. The Moringa tree, known to early civilizations as the tree of life, because of its ability to thrive where other plants withered. And from the beginning its story was tied not only to survival but to the idea of hidden potential stored deep within its fibrous bark, oily seeds, and lush green leaves. Potential that communities across ancient India, particularly along the Ganges basin, recognized when they used its bark in poultices, its leaves in food, and its oil for both lamps and skin remedies, creating a tradition of practical reverence for a tree that was unlike any other. The ancient texts of Ayurveda, which emerged around 2000 BCE, refer to Moringa as Shigru, a healer's ally that could cool fevers, reduce swelling, and sharpen alertness. And these early medicinal uses provided not just relief to the sick but an enduring narrative thread, tying Moringa to cultural identity, ritual healing, and daily sustenance, so much so that the tree's presence near villages became almost symbolic of health itself, a living pharmacy whose roots were seen as capable of balancing energies within the body according to the principles of dashas, an idea that may not have understood the genetic underpinnings of plant chemistry, but instinctively aligned with Moringa's biochemical richness that we now know includes a staggering range of antioxidants, vitamins, and amino acids. Meanwhile, outside the Indian plains, ancient travelers and merchants carried seeds across trade routes that linked South Asia with the Arabian Peninsula and eventually Africa, where the plant adapted once more to arid soils, embedding itself into local traditions such as Ethiopian herbal medicine and Egyptian rituals where oil from the seeds was prized as an ointment and preservative for the skin of both the living and the embalmed. Signaling that long before laboratories ever looked at its DNA, Moringa had established itself as a bridge between nourishment and spiritual continuity in civilizations that regarded plants not only as food but as guardians of vitality and endurance. Archaeological findings from around the Nile Valley suggest that Moringa oil jars were placed in tombs alongside gold and papyrus, implying that this tree's value transcended mere utility, carrying instead a mark of permanence, resilience, and healing power strong enough to accompany a soul into the afterlife. And while we cannot know if ancient Egyptians speculated about the microscopic blueprints encoded within its cells, their preservation practices hint at an intuitive recognition of the plant's unique chemistry, since the oil resisted rancidity longer than many others, a property which modern science attributes to its uncommon combination of fatty acids and stabilizing compounds, once again showing how practice preceded explanation by thousands of years. In the Greco-Roman world, Moringa made its appearance through maritime trade, arriving as a curiosity but swiftly entering the lexicon of natural remedies, with physicians like Dioscorides noting its value as an extract for inflammation, and Pliny the Elder describing its oil as a cleansing agent, evidence that the tree's reputation was expanding across cultures and geographies, being shaped and reshaped by each society that adopted it, embedding local meanings while retaining its core identity as a healer's plant, a plant that seemed to adapt as much socially as it did biologically, finding roots in Mediterranean gardens and Arab markets just as easily as in the soil of South Asia. This historical adaptability matters because it points toward a deeper truth about Moringa's nature. It is a survivor, a species with genetic resilience encoded into its very DNA, enabling it to flourish in poor soils, withstand drought, and rebound after harsh seasons, a trait that human communities interpreted as vitality and strength, but which modern genomics interprets as the product of evolutionary adaptations at the molecular level, including stress response genes and secondary metabolites that shield the plant from environmental extremes. Genes that in turn became of interest to scientists not only for agricultural purposes but eventually for medical exploration, since compounds that protect plants often have potent effects on human physiology as well. Yet before science dissected genes, Moringa's story unfolded in myth, ritual, and subsistence, whether in Indian epics that praise its vitality, African folk practices that employed its leaves in fertility rites, or Middle Eastern traditions where its oil was rubbed onto the skin of wrestlers and warriors, reinforcing a cultural idea that this tree was more than just sustenance. It was a force that could enhance, protect, and sustain human strength, a belief that framed centuries of usage and ensured that Moringa never faded into obscurity even as empires rose and fell around it. By the medieval period, Islamic scholars such as Avicenna catalogued Moringa's properties in the canon of medicine, which became a cornerstone for both European and Middle Eastern medical knowledge, carrying forward the accumulated traditions of India, Africa, and the Arab world into a framework that blended observation with philosophy, ensuring that Moringa's reputation as a medicinal tree did not remain confined to local lore but entered scholarly discourse where it could be debated, refined, and preserved for future generations, a crucial step that kept the plant alive in the intellectual memory of medicine long before its molecular secrets would be revealed. From this ancient foundation of reverence, use, and exchange, the Moringa tree carried with it a centuries-old legacy, a story of survival, of people recognizing value long before they could explain it, and of cultures weaving it into their fabric of health, all of which set the stage for the modern scientific journey that would one day pierce beyond the surface of leaves and seeds to explore the genetic code beneath, asking not only what Moringa could do as a plant but what its DNA might reveal about medicine, biology, and the shared resilience of life itself. Part 2. Early Herbal Traditions and Healing Uses. As the Moringa tree spread from its ancient roots into broader cultural landscapes, it was absorbed into the herbal traditions of diverse societies, each layering new meanings upon its already storied existence. And in these traditions, Moringa was not simply viewed as a tree but as a living dispensary of remedies that addressed the fundamental ailments of human life, from hunger to infection to fatigue, creating a legacy that linked community well-being directly to the natural world. In India, where Ayurvedic medicine had already elevated Moringa into the ranks of essential healing plants, practitioners developed elaborate preparations that incorporated its roots, bark, and leaves into decoctions meant to balance the body's internal energies, prescribing it for digestive complaints, joint pain, skin conditions, and even as a tonic for sharpening the senses. A breadth of applications that reflected both empirical trial and an intuitive grasp of the plant's wide-ranging chemical activity, a fact modern pharmacology would much later confirm by isolating active compounds such as quercetin, isothiocyanates, and chlorogenic acid, all known to exert effects on inflammation and oxidative stress. In Africa, where the tree adapted to varied climates and became embedded in local traditions, healers used Moringa leaves in teas for stomach ailments, seeds as a purifying agent for drinking water, and bark and poultices applied to wounds, showcasing not only medicinal but hygienic uses that aligned with broader communal needs, and this emphasis on purification hinted at a recognition of Moringa's antibacterial properties, since even without microscopes communities observed that water treated with crushed Moringa seeds was clearer and safer to drink, a practice which centuries later would be studied by researchers seeking sustainable water purification methods. In the Middle East, where Moringa was carried along trade routes, its oil became associated with both medicinal and cosmetic practices, massaged into aching joints, rubbed on skin to prevent dryness, and burned as lamp oil in rituals that connected light and health, with early physicians noting that the oil resisted spoiling and seemed to protect the skin from harsh desert conditions, an observation that underscores how practicality and spirituality often overlapped in herbal traditions, with plants serving both as physical medicine and symbolic protection. In Southeast Asia, Moringa adapted into tropical soils and became a household plant, its young pods cooked in curries, its leaves used in soups, and its roots in traditional remedies for circulatory health, embedding it not only in healing but in cuisine, where daily use reinforced its role as preventive medicine, a principle still central to many herbal systems where the line between food and medicine is blurred, suggesting that Moringa's significance was as much about sustaining vitality as treating illness. This widespread integration into daily life demonstrates how herbal traditions preserved Moringa's status across generations, with knowledge transmitted orally from healer to apprentice, and mothers teaching daughters how to prepare nourishing broths from the leaves for the sick, embedding the plant into social memory in ways that ensured it could never be forgotten, even during periods of cultural upheaval or famine, because the tree itself survived droughts and harsh conditions, providing a constant source of sustenance and healing when other plants failed, a resilience that mirrored the continuity of knowledge about its uses. What is striking when comparing these herbal traditions across regions is the consistency of themes: purification, nourishment, inflammation, vitality, all recurring motifs that suggest human observers, without knowing biochemistry, nonetheless identified the same therapeutic potential through careful observation, repetition, and communal wisdom, and this convergence of knowledge across distant cultures hints that Moringa's pharmacological impact is not incidental but deeply rooted in its unique chemical and genetic composition, which consistently produced observable effects strong enough to be recognized independently by societies separated by geography and language. Yet these traditions were never static, as new ailments and challenges reshaped how Moringa was applied, with healers modifying recipes to suit local conditions, sometimes blending Moringa with other herbs to enhance its effects or to mask its slightly bitter flavor, thereby creating complex pharmacopoeias that demonstrate early human ingenuity in maximizing the potential of natural resources, ingenuity that modern pharmacognosy studies in detail when attempting to replicate or understand ancient remedies. In this context, Moringa's role in herbal medicine was not simply about curing disease but also about sustaining communities against uncertainty, because when drought struck, its leaves could still be harvested for food, and when epidemics emerged, its extracts offered relief for symptoms, serving as both shield and sustenance, embodying a dual role that positioned it as indispensable across centuries, a tree that could address both the body's need for nutrients and its battles against illness. By the medieval period, when global exchanges of medical knowledge intensified through the translation movements in Baghdad, Cordoba, and later into Europe, Moringa's descriptions began to circulate in scholarly texts alongside those of other renowned medicinal plants, ensuring its reputation was preserved not only in oral traditions but also in written medical literature that influenced practitioners from Arabia to Italy, a step that allowed its uses to persist even as societies transformed, making it part of a broader conversation about the natural world's role in healing. And while much of the emphasis in these texts was descriptive, the very act of cataloging ensured that Moringa entered the intellectual record of humanity's quest for health. Throughout these centuries, one can see how the threads of local tradition, practical observation, and scholarly preservation intertwine to weave a continuous narrative of Moringa as a healer's ally, and this narrative, though lacking in modern scientific terminology, represents an unbroken chain of knowledge that bridged generations, cultures, and geographies, establishing a foundation strong enough that when modern science finally turned its attention to Moringa's DNA, it was building upon centuries of lived experience that had already confirmed the plant's value in practice if not in molecular explanation, a reminder that tradition often anticipated discovery, and that the wisdom of herbal medicine carried within it the seeds of future breakthroughs. Part 3. The Botanical Mystery of a Resilient Tree. While Moringa's presence in herbal traditions and healing lore laid the groundwork for its cultural significance, the tree itself carried mysteries that fascinated observers long before modern genetics could unravel them. For here was a species that seemed to defy environmental limitations, thriving in poor soils where crops withered, enduring long dry seasons, and regenerating even after being cut down to its trunk. A resilience so striking that it shaped both how people valued the tree and how they interpreted its symbolic meaning. In regions of Africa, for example, villages saw Moringa as a tree that could not die, a provider that returned season after season with edible leaves even when famine threatened, and this observation turned into mythic narratives where Moringa was associated with renewal and immortality, stories that made sense to communities who depended on its persistence during hardship. While in India, where sacred groves often incorporated medicinal plants, Moringa's capacity for rapid regrowth marked it as a living emblem of continuity, an idea reinforced by its constant availability as food and medicine even in lean times. Botanically, what made Moringa unusual was not only its adaptability but its astonishing speed of growth, with seedlings maturing into trees capable of flowering and fruiting within a year, a cycle that puzzled early agriculturalists who noted how few plants could convert sunlight, water, and poor soil nutrients into biomass so efficiently, and though they lacked the language of chloroplast density, nutrient uptake, or genetic expression, their recognition of Moringa's productivity meant it was cultivated wherever survival demanded reliability, reinforcing its reputation as a miracle tree. Scientifically, this growth pattern hinted at unique genetic pathways controlling photosynthesis and stress responses, traits that biologists centuries later would identify as linked to a suite of genes regulating stomatal conductance, water efficiency, and protein synthesis. But for ancient farmers these traits were visible only in the practical sense. Moringa thrived when nothing else could, a fact that elevated it from useful plant to essential lifeline. The tree's morphology added to its mystery, because every part seemed to offer something valuable. Leaves rich in nutrients, pods edible and palatable, seeds producing oil, bark yielding medicinal extracts, and roots carrying compounds strong enough to be used sparingly in treatments, creating an impression of a plant that was complete in itself, designed by nature to sustain life in multiple dimensions. And this wholeness distinguished it from other flora that offered only one or two uses, ensuring that communities never abandoned Moringa even when other food or medicinal plants fell out of favor. Its ecological role further underscored its uniqueness, for Moringa not only survived but often improved the soil around it by dropping nutrient-rich leaves that decompose quickly, replenishing organic matter, and by casting light rather than dense shade, allowing intercropping with grains and legumes, an agricultural synergy that farmers could observe empirically, using Moringa as a companion tree long before agronomists would articulate concepts of nitrogen cycling and ecosystem balance, demonstrating once again how lived practice anticipated scientific validation. In landscapes prone to drought, Moringa's deep taproot allowed it to access water hidden beyond the reach of shallow-rooted plants, ensuring that its leaves remained available even during dry seasons. And this underground resilience made it particularly valued by pastoralists and farmers who recognized that Moringa was dependable in ways that other vegetation was not, leading to its incorporation not just into medicine and diet but into livestock care, as animals fed on Moringa leaves retained strength, milk production, and fertility even when forage was scarce, thus embedding the tree's role into economic and survival systems that extended well beyond individual health. To the observer, all of these traits combined to make Moringa a botanical mystery, a plant that seemed to hold more vitality than its modest size suggested, and this aura of vitality inspired not only practical reverence but also speculation, with some traditions treating Moringa as a plant blessed by divine forces, while others saw it as a natural teacher, showing humans how resilience and generosity could coexist. And though modern science strips away such mythic layers, the fascination remains, because in many ways the genetic explanations now being discovered only deepen the wonder by revealing how precisely tuned the tree's molecular machinery is for survival and abundance. This dual identity, as both a mystical emblem in traditional narratives and a subject of scientific curiosity in modern laboratories, defines Moringa's journey through history, bridging worlds of knowledge that often seem opposed but in this case complement each other. For the tree's visible resilience invited stories while its invisible resilience, encoded in DNA, awaited discovery, and together these perspectives underscore the enduring human impulse to make sense of extraordinary phenomena through both imagination and investigation. The mystery, then, was not simply that Moringa could survive, but that it seemed to give more than survival required, producing leaves rich in protein, vitamins, and minerals far beyond what was typical for trees in similar environments, almost as if nature had concentrated abundance into a single species, a generosity that traditional healers described in spiritual language but which modern nutritionists quantify in biochemical tables showing levels of iron, calcium, vitamin A, and essential amino acids that rival or exceed conventional food sources. A convergence that once more illustrates how empirical observation has always walked hand in hand with later scientific explanation. What began as a tree that stood out for its toughness became, in the eyes of those who used it, a puzzle of generosity, a plant that gave too much, too reliably, too abundantly, and it was this sense of excess that made it legendary, ensuring its persistence not only in ecosystems but in cultural memory, so that by the time genetics matured as a discipline in the 20th century, researchers would inherit not just a plant to study but a mystery to solve, one framed by millennia of observation, reverence, and reliance, ready to be unlocked not by speculation but by sequencing, mapping, and decoding the genetic script that had allowed Moringa to thrive where other plants fell silent. Part 4. Colonial Encounters and Scientific Curiosity. When European colonial expansion spread across Asia, Africa, and the Middle East between the 16th and 19th centuries, countless plants encountered new scrutiny through the lens of Western science, and among them the Moringa tree quickly caught attention, not merely because of its resilience in harsh climates but because of the multiplicity of uses that local populations attributed to it, a breadth of claims that intrigued colonial physicians, naturalists, and explorers who sought to catalog and classify the flora of conquered lands. In British India, where Moringa had long been embedded in Ayurvedic traditions, colonial medical officers stationed in cantonments noted the plant's popularity among local healers, who prescribed its leaves for fevers, its pods as nourishing vegetables, and its seeds as both medicine and oil sources, leading to early reports being sent back to institutions like the Royal Society in London, which at that time was eager to gather specimens from across the empire for botanical gardens such as Kew, thereby transforming Moringa from a local remedy into a subject of global botanical record. The act of cataloging, however, often stripped plants from their cultural contexts, so while British records described Moringa in terms of Latin binomials and morphological features, they frequently overlooked or dismissed indigenous knowledge systems, regarding them as anecdotal rather than scientific. Yet paradoxically, these very notes about local uses guided further European investigations, highlighting an irony of the colonial encounter. Western science both dismissed and depended upon indigenous knowledge at the same time. French colonial botanists in North Africa also encountered Moringa and remarked on its drought resistance, particularly noting its ability to grow in marginal soils where attempts to cultivate European crops had failed. And in their reports, the tree was often framed as a potential agricultural resource for colonial economies, showing how scientific curiosity was intertwined with imperial ambition, as the search for resilient crops was less about indigenous well-being and more about sustaining military posts, plantations, and settlement projects. Meanwhile, in the Caribbean, where enslaved Africans carried with them both seeds and memories of Moringa's healing potential, the tree established itself once again as both food and medicine, and colonial physicians took note of its uses among enslaved populations, sometimes appropriating those remedies into plantation medical kits, reinforcing the tree's reputation as a global migrant that followed human movement across oceans, adapting wherever people transplanted it, and sustaining lives under both free and forced circumstances. During the 18th and 19th centuries, the rise of pharmacology as a scientific discipline also encouraged experiments with Moringa extracts, as European chemists attempted to isolate active compounds. That might explain the plant's reputed benefits, though these early studies lacked the precision of later biochemistry, and often results were inconclusive or contradictory, leading some journals to relegate Moringa to the category of curiosities, while others insisted it deserved further attention, thereby cementing its place in the gray zone between folk remedy and scientific substance, awaiting more sophisticated methods to reveal its secrets. The oil extracted from Moringa seeds, known as Ben oil in colonial trade records, became particularly valued for its clarity, lack of rancidity, and usefulness in lubrication and perfumery, leading to small but steady commerce that brought the tree into European markets, and this commercial angle reinforced curiosity among chemists, who recognized that such stability in oil must have a chemical explanation, though the molecular structures of fatty acids would not be mapped until the late 19th century, leaving Moringa's oil as both a practical export and a chemical enigma. Botanical gardens in Europe, from Kew to Paris, cultivated Moringa specimens alongside other exotic plants, using them as teaching tools for students of medicine and botany, which meant that for the first time in history, Moringa was observed not in the context of its native environment but under controlled European conditions, where its fast growth and unusual adaptability impressed horticulturists, who recorded that few other trees could thrive so readily when transplanted across climates, reinforcing earlier observations from Africa and India but now framed as data within the expanding field of comparative botany. This colonial-era curiosity also intersected with missionary activity, as Christian missionaries often learned about Moringa through their work in villages, sometimes adopting it into their own dispensaries when treating local populations, while simultaneously documenting its uses in letters and reports that circulated within European societies, further embedding Moringa into the collective archive of plants that might hold promise for the future, though still lacking the scientific frameworks to fully decode why it was so effective. What emerges from this period is a layered picture. Moringa as a plant of survival for indigenous peoples, Moringa as a curiosity for scientists, Moringa as a commodity for traders, and Moringa as a tool of adaptation for colonizers struggling to master unfamiliar landscapes, all overlapping in ways that ensured its continued global spread, while also laying the groundwork for the eventual transition into modern science, where genetics and molecular biology would provide the explanations that colonial observers lacked. Yet it is important to note that colonial encounters also distorted the narrative of Moringa, often framing it as something discovered by Europeans despite centuries of indigenous use, a rhetorical erasure that obscured the deep traditions which had preserved the plant's role in medicine for millennia, and this pattern repeated across many colonial contexts, where indigenous knowledge was mined for resources but rarely credited as foundational science, a legacy that modern researchers are only now beginning to acknowledge as they revisit plants like Moringa with fresh respect for traditional ecological knowledge. Thus, the colonial period did not reveal the genetic mysteries of Moringa, but it set the stage for their eventual exploration. By circulating the tree across the globe, embedding it in scientific archives, and piquing curiosity about its remarkable properties, ensuring that when the tools of molecular biology finally emerged in the 20th century, Moringa was already recognized as a plant worth studying, a plant that had traveled from sacred groves to imperial gardens, from healing rituals to laboratory benches, carrying with it the weight of survival, adaptation, and unresolved mystery that made it an irresistible subject for the next stage of scientific inquiry. Part 5. Global Spread of a Medicinal Wonder. As the colonial period carried Moringa into botanical gardens, trade networks, and medical archives, the 19th and early 20th centuries witnessed an acceleration of its spread across continents, transforming it from a regional healer's ally into a plant with global recognition, a process driven not only by colonial curiosity but also by the practical reality that wherever it was planted, Moringa thrived and offered sustenance, making it indispensable to communities struggling with hunger, poverty, and health challenges. And this global migration created a new stage in its history where local traditions began to overlap with international exchanges, embedding Moringa into a web of uses that stretched from Asia to the Americas. In Africa, where Moringa had already been adopted centuries earlier, its importance deepened as colonial agricultural programs sought resilient species to withstand cycles of drought. And though such programs often prioritized cash crops, local farmers continued cultivating Moringa in household gardens, ensuring its leaves, seeds, and pods remained part of everyday diets and remedies, a quiet persistence that kept it alive even when colonial policies disrupted food systems. And in places like Sudan and Ethiopia, Moringa trees became fixtures near villages and wells, symbolizing continuity amid change, providing both shade and nutrition for families and livestock. In South Asia, particularly India and Sri Lanka, Moringa's role expanded from traditional medicine into mainstream cuisine, with drumstick pods becoming a staple in curries, soups, and stews, embedding the plant so deeply into diet that it no longer functioned only as a healer but as an everyday source of nutrients, a transition that blurred the lines between medicine and food, reinforcing its preventive role in health. And as migrants from the Indian subcontinent traveled during the 19th century to work in plantations across the Caribbean, East Africa, and Southeast Asia, they carried Moringa seeds and recipes with them, effectively transplanting both the plant and the knowledge of its uses into new landscapes where it took root once more. In the Caribbean, where enslaved Africans had already introduced Moringa centuries earlier, indentured laborers from India reinforced its presence, adding new culinary traditions that layered upon existing African uses. And by the 20th century, Moringa had become so common across islands like Jamaica, Trinidad, and Martinique that it was often mistaken as a native species, its leaves used in teas for colds and fevers, its pods eaten as vegetables, and its seeds valued for water purification, demonstrating how cultural blending through migration ensured Moringa's global spread while adapting it to local needs. In Latin America, particularly in Mexico, Central America, and Brazil, Moringa gained attention in the 20th century as agricultural organizations promoted it as a high-protein fodder for livestock and a nutritional supplement for impoverished communities, embedding it into programs that sought to combat malnutrition, especially among children. And here the global spread intersected with humanitarian efforts, as NGOs and international agencies recognized that Moringa's fast growth, resilience, and dense nutrition made it an ideal crop for regions suffering food insecurity, further reinforcing its reputation as the miracle tree. Meanwhile, in the Philippines and Southeast Asia, Moringa, locally called malunggay, became central to daily diets and was recognized for its role in promoting maternal health, with leaves traditionally given to breastfeeding mothers to boost milk production, a practice later studied by medical researchers who confirmed its galactagogue properties, illustrating once more how local traditions anticipated scientific validation. And this emphasis on maternal and child health contributed to Moringa's enduring cultural importance in the region, ensuring that its spread was not only geographic but generational, passed down through familial and community practices. What made Moringa's global spread remarkable was not simply its physical transplantation but the way it integrated into new cultural fabrics, absorbing local meanings while retaining its identity as a plant of abundance and healing. So that whether it was known as shigru in India, malunggay in the Philippines, benzoolive in Haiti, or drumstick tree in English colonies, it carried with it a core narrative of resilience, generosity, and health, adapted yet consistent, local yet universal, a rare example of a plant whose uses were so versatile that no matter the culture, it found a place both on the plate and in the medicine chest. As the 20th century advanced, the scientific community increasingly noticed this widespread adoption, and researchers began to ask why so many unrelated societies had converged on similar uses for Moringa, a question that hinted at deeper biochemical explanations that would only be unraveled through molecular biology. But even before DNA analysis became possible, nutritional studies in the 1970s and 1980s confirmed that Moringa leaves contained exceptionally high levels of protein, iron, calcium, and vitamins, explaining why it had been such a consistent tool against malnutrition throughout history. And this validation gave further momentum to development programs that planted millions of Moringa trees in Africa, Asia, and Latin America as part of international health strategies. The global spread also brought Moringa into popular culture, where it was increasingly marketed in health stores and wellness circles as a superfood, a term that captured its dense nutrient profile but often obscured the centuries of indigenous knowledge that had preserved its use, raising questions about appropriation, recognition, and the balance between commercialization and respect for cultural heritage. Yet, regardless of such debates, the very fact that Moringa could be consumed as powder in a smoothie in New York while still being boiled into a village soup in rural India demonstrated the breadth of its global reach, a reach that few plants in human history have achieved. Ultimately, this period of global spread represents not only the movement of a plant across borders but the merging of traditions, the layering of uses, and the convergence of human ingenuity in recognizing value where it existed, ensuring that by the time genetic research entered the stage, Moringa was not an obscure tree awaiting discovery but a globally embedded organism whose nutritional and medicinal reputation was already secure, waiting only for science to explain at the molecular level what cultures across continents had always known through lived experience. In this sense, The spread of Moringa set the stage for a transformation in how it would be perceived, because while earlier ages saw it as healer, survivor, or food source, the modern age, armed with genetic tools, would come to see it as a subject of molecular wonder, a living archive of evolutionary strategies, encoded in DNA, that might unlock not just explanations for its resilience but pathways for revolutionizing medicine itself. Part 6. The Rise of Molecular Biology in Plant Research. The 20th century witnessed a profound transformation in how humanity studied plants, for the focus shifted from simply observing visible traits to probing the molecular machinery that governed growth, adaptation, and chemical production. And within this new framework of molecular biology, Moringa emerged not merely as a curiosity from herbal traditions, but as a promising subject whose extraordinary resilience and nutritional density seemed to demand deeper genetic explanation. The roots of this shift can be traced back to the mid-1900s, when the discovery of DNA's double helix structure in 1953 by James Watson and Francis Crick, alongside the critical X-ray crystallography work of Rosalind Franklin, laid the foundation for understanding heredity at the molecular level, a breakthrough that revolutionized every field of biology by revealing that life itself was coded in sequences of nucleotides. And from that moment onward, plants were no longer studied solely in terms of morphology or physiology, but as repositories of genetic information that could be mapped, compared, and manipulated. In agriculture, this meant the Green Revolution of the 1960s and 1970s not only introduced new crop varieties but also triggered efforts to understand the genetic basis of yield, disease resistance, and nutrient content. And while the initial focus was on staple crops like wheat, rice, and maize, the methods being developed, from electrophoresis to molecular markers, would soon extend to lesser-known plants, including Moringa, whose unusual traits made it an attractive candidate for study once research infrastructures expanded in Asia, Africa, and Latin America. The rise of molecular biology also coincided with advances in phytochemistry, as scientists began isolating active compounds from medicinal plants and linking their production to genetic pathways. So while ancient herbalists described Moringa as anti-inflammatory, modern researchers sought to identify which genes regulated the biosynthesis of isothiocyanates or flavonoids responsible for such effects, thereby merging traditional observations with molecular explanation in ways that validated indigenous knowledge while pushing it into new scientific frontiers. In the 1980s and 1990s, the development of polymerase chain reaction (PCR) technology allowed scientists to amplify specific DNA segments, making it possible to study genes even in plants that lacked full genome sequences, and this technique opened the door for early investigations into moringa's genetic diversity, enabling researchers to compare varieties across different regions and climates, documenting how the tree adapted so effectively to drought or poor soils, and raising the possibility that its genome contained unique stress response elements of great value to both agriculture and medicine. The parallel rise of biotechnology further deepened this interest, for as scientists learned to transfer genes between organisms, creating genetically modified crops resistant to pests or herbicides, they began to look at plants like Moringa not only as subjects of study but also as potential sources of valuable genes, imagining futures where moringa's resilience traits could be introduced into food staples, or where its medicinal compounds could be enhanced through genetic engineering, a vision that was speculative at the time but rooted in the growing confidence that DNA was a code that humanity could read and rewrite. At the same time, plant molecular biology was increasingly shaped by global health concerns, as malnutrition and chronic diseases demanded innovative solutions. And in this context, moringa's unusually high nutritional content drew attention from researchers funded by international development agencies, who hoped that by unlocking the genetic basis of its nutrient density, they might engineer similar traits into widely consumed crops, creating a form of biofortification that could address vitamin and protein deficiencies on a global scale. Moringa's case was particularly compelling because unlike many other medicinal plants whose active compounds were rare or toxic, its leaves, pods, and seeds were safe for regular consumption, meaning that any genetic insights could be directly applied in both nutritional and therapeutic contexts, making it a bridge between food science and medicine, a duality that matched perfectly with the interdisciplinary spirit of molecular biology's rise. Where boundaries between fields were increasingly blurred in pursuit of holistic understanding, the rise of molecular plant research also fostered collaborations between developed and developing nations, with universities in India, the Philippines, and Africa partnering with institutions in Europe and North America to exchange genetic samples, conduct joint studies, and train young scientists in techniques like DNA sequencing and tissue culture. And Moringa often featured in these projects because it was locally abundant, culturally significant, and scientifically intriguing, offering students a tangible subject where modern tools could be applied to questions of both local and global relevance. This period also marked a philosophical shift in how plants were perceived, as molecular biology emphasized that resilience, nutrition, and healing properties were not magical or mysterious but encoded traits shaped by evolution. And in Moringa's case, the quest became one of deciphering how its genome orchestrated the production of such a wide range of beneficial compounds while also enabling survival in harsh climates, a dual genetic strategy that scientists suspected might hold lessons for both agriculture and medicine. By the late 20th century, the sequencing of model plant genomes like Arabidopsis thaliana set a precedent for studying less prominent species. And although Moringa did not yet have a fully sequenced genome, preliminary work on its DNA markers and genetic diversity laid the groundwork for the 21st century, when next-generation sequencing technologies would finally unlock its genetic blueprint, an achievement that promised not only to confirm centuries of traditional knowledge but also to reveal entirely new possibilities for medical innovation. In hindsight, the rise of molecular biology did not immediately revolutionize how Moringa was used, but it fundamentally changed the questions being asked about it, shifting the focus from what the tree could do in practice to why it could do those things at the molecular level, setting the stage for the next chapter in its story where the secrets hidden in its genome would begin to be revealed with unprecedented clarity, transforming Moringa from a plant of tradition and resilience into a subject of cutting-edge genetic exploration with the potential to reshape the very future of medicine. Part 7. Mapping Moringa's Genetic Blueprint. The dawn of the 21st century brought with it a revolution in genetic research, for advances in sequencing technologies made it possible to read entire genomes at speeds and costs unimaginable only a decade earlier. And in this new era of genomics, Moringa, long cherished for its medicinal and nutritional virtues, entered a transformative phase where its genetic blueprint could finally be decoded in unprecedented detail. The earliest steps toward mapping Moringa's genome began with the construction of genetic libraries, where researchers carefully extracted DNA from its leaves and seeds, fragmented it into smaller pieces, and fed it into sequencing machines that translated the nucleotide code into long strings of data, a process that required both sophisticated laboratory tools and powerful computational systems capable of aligning, assembling, and interpreting millions of fragments to form a coherent representation of the plant's genetic architecture. This work was challenging, for unlike model organisms such as Arabidopsis thaliana or rice, Moringa lacked a long history of genetic study, meaning that researchers had to start from scratch, developing reference points, designing primers for amplification, and cross-checking results across multiple samples to ensure accuracy, a laborious process that nevertheless revealed the first glimpses of Moringa's remarkable genomic complexity. Early findings confirmed what traditional knowledge had long suggested. Moringa's genome was rich in genes associated with secondary metabolite pathways, including those responsible for producing antioxidants, anti-inflammatory compounds, and bioactive phytochemicals, and these discoveries provided molecular confirmation of centuries of herbal practices, demonstrating that Moringa's healing properties were not anecdotal myths but the result of genetic programs evolved over millennia to protect the tree and, by extension, benefit humans who consumed it. Sequencing also uncovered genes linked to drought tolerance, high nutrient accumulation, and pest resistance, traits that explained why Moringa thrived in marginal soils and arid climates where other plants withered. And scientists quickly recognized that these genetic features were not only valuable for understanding Moringa itself but also held immense potential for crop improvement programs worldwide, since transferring or mimicking such resilience traits in food staples could strengthen food security. In an era of climate change, the mapping process illuminated another striking aspect of Moringa: its unusually efficient regulation of protein synthesis in its leaves, a feature rare among plants, which contributed to its high nutritional value, and this led molecular biologists to investigate promoter regions in its DNA that might be responsible for activating nutrient-rich pathways, opening the possibility of using these genetic switches to enhance other crops or even to engineer functional foods designed to combat malnutrition. Yet mapping Moringa's genome was not simply about cataloging genes. It was also about identifying unique regulatory networks, the orchestration of transcription factors and epigenetic markers that determined when and how compounds were produced, and in this regard Moringa stood out as a genetic orchestra conductor, fine-tuning the production of bioactive compounds in response to environmental stresses, an adaptive brilliance that scientists hope to harness for medical applications, particularly in designing drugs that could mimic these natural stress response strategies. The sequencing efforts further revealed high genetic diversity across different Moringa species and populations, with varieties from Africa, South Asia, and Latin America each possessing subtle genomic variations that influenced nutrient content and medicinal potency, and this diversity became a treasure trove for researchers, suggesting that selective breeding or Biotechnological modification could combine the best traits from multiple lineages to create optimized versions of Moringa tailored for specific medical or nutritional uses. The publication of Moringa's draft genome sequence in scientific journals sparked a surge of interest, drawing attention from pharmaceutical companies, agricultural institutions, and global health organizations eager to explore the plant's potential. And this convergence of interests highlighted how the simple act of mapping DNA could transform a traditional herbal remedy into a modern scientific asset, a shift that carried both promises and challenges in terms of ownership, access, and ethical use. Beyond health and agriculture, genomic mapping also opened doors for ecological research, for by comparing Moringa's genetic strategies with those of other drought-tolerant plants, scientists began to piece together broader insights into how vegetation adapts to climate extremes, knowledge that could inform conservation strategies and sustainable farming practices, ensuring that Moringa's story was not only about human benefit but also about understanding the intricate dialogue between plants and their environments. The mapping process itself was emblematic of 21st-century science, involving international collaborations across continents, with researchers in India sequencing raw DNA, bioinformatics teams in Europe assembling the data, and agricultural scientists in Africa validating findings through field studies, a global mosaic of expertise that underscored how a tree native to South Asia and Africa had become a symbol of interconnected scientific ambition, uniting diverse cultures in pursuit of knowledge encoded in a humble leaf. As the genome map became more refined with the aid of next-generation sequencing and CRISPR-based annotation tools, scientists began to identify candidate genes for therapeutic exploration, such as those involved in the biosynthesis of niazimucin, a compound with anti-cancer potential, or moringin, a molecule linked to anti-inflammatory activity, and by tracing the genetic origins of these compounds, researchers could imagine designing synthetic pathways in laboratories or enhancing natural production in cultivated Moringa varieties, effectively bridging the gap between raw herbal remedies and standardized pharmaceutical applications. In reflecting on this achievement, one sees that the mapping of Moringa's genetic blueprint was not an endpoint but a new beginning, for it transformed the plant from a subject of folklore and nutrition into a databank of molecular strategies with applications ranging from medicine to agriculture. And by giving humanity the tools to read Moringa's code, genomics unlocked a future where its healing power could be studied, modified, and expanded with scientific precision, setting the stage for the next great leap, decoding not just which genes Moringa possessed, but how its proteins functioned in shaping life-saving compounds that might redefine the possibilities of modern medicine. Part 8. Decoding the secrets of Moringa's proteins. With a mapping of Moringa's genome providing a detailed inventory of its genetic material, the next frontier for researchers became the functional exploration of its proteins. For while DNA provides the blueprint, it is proteins that execute the instructions of life, shaping metabolism, immunity, and healing responses, and in the case of Moringa, its remarkable medicinal and nutritional properties could only be fully understood by decoding the unique proteins and enzymes that its genome encoded. The journey into Moringa's proteome began with extraction and purification of proteins from different tissues, including leaves, seeds, and pods, each of which contained specialized molecules adapted to distinct functions. And scientists soon discovered that Moringa's leaves were particularly rich in proteins with antioxidant properties, capable of neutralizing free radicals and reducing oxidative stress in human cells, a finding that explained its traditional use in strengthening vitality and combating fatigue. Among the most striking proteins identified were those with antimicrobial activity, small peptides capable of disrupting bacterial membranes or inhibiting the growth of pathogens, and laboratory assays confirmed that these molecules could act as natural antibiotics, suggesting that Moringa had evolved a powerful biochemical defense system against infections, which humans had unknowingly harnessed through centuries of herbal medicine. Attention also turned to Moringa's seeds, which produced proteins involved in water purification. For certain cationic proteins derived from seed extracts were shown to flocculate impurities and neutralize harmful microbes, a property that had been used in traditional communities for centuries to clean drinking water, but which molecular biology now revealed to be the result of highly specialized protein structures with remarkable binding capacities, opening possibilities for low-cost bioengineered water purification technologies. Further studies revealed enzymes in Moringa that were unusually efficient at breaking down complex compounds, including detoxifying phytochemicals that could otherwise be harmful, and these enzymes drew interest from pharmacologists who saw potential applications in designing therapies for metabolic disorders or drug detoxification processes in the human body, thereby extending Moringa's relevance from nutrition into pharmacokinetics and drug development. But perhaps the most exciting discoveries emerged from investigations into bioactive proteins involved in signaling pathways. For For Moringa was found to contain proteins that could influence inflammatory cascades by modulating cytokine activity, effectively dampening harmful immune responses while enhancing protective ones, a dual action that scientists considered particularly valuable for treating conditions such as arthritis, autoimmune disorders, and even certain cancers where immune regulation was key. In addition, structural biology techniques like X-ray crystallography and cryo-electron microscopy allowed researchers to visualize Moringa's proteins at atomic resolution, revealing unique folds and motifs not commonly found in other plants, structural peculiarities that explained why these proteins had such potent bioactivities and which offered templates for synthetic drug design. 4 chemists could now mimic or modify these natural scaffolds to create novel therapeutics inspired by Moringa's molecular ingenuity. The study of Moringa's proteome also intersected with nutritional science, as researchers identified proteins in its leaves that contributed to its unusually high amino acid content, including essential amino acids often lacking in vegetarian diets, a finding that reinforced Moringa's role as a complete food and explained its success in combating malnutrition in communities where animal protein was scarce, while also raising interest in using Moringa-based protein supplements for global health interventions. Proteomics also highlighted the dynamic nature of Moringa's proteins, for their expression patterns shifted dramatically in response to environmental stresses such as drought, heat, or pest attack, and this plasticity suggested that Moringa's genome encoded a highly adaptable protein network designed for survival in hostile conditions. Insights that could guide crop engineering projects aimed at creating stress-resilient food systems for a changing climate. An intriguing discovery involved storage proteins in Moringa seeds that displayed anti-carcinogenic properties, with laboratory tests indicating that they could trigger apoptosis in certain cancer cell lines. And while clinical applications remained far in the future, these findings hinted that Moringa's proteins might one day form the basis of novel cancer therapies that were less toxic and more targeted than conventional chemotherapy. Equally promising were proteins associated with neuroprotection, as preliminary studies suggested that certain Moringa-derived peptides could cross the blood-brain barrier and shield neurons from oxidative stress, raising the possibility that the plant might offer leads for treating neurodegenerative diseases like Alzheimer's or Parkinson's, conditions for which current therapies remain limited and often ineffective. As the proteomic catalog grew, scientists began integrating this data with genomic and metabolomic information, creating a systems biology view of Moringa where DNA, proteins, and chemical compounds were understood as parts of a unified network, a holistic approach that reflected how the plant's medicinal effects arose not from isolated molecules but from the coordinated actions of complex biochemical pathways, an insight that pushed research away from reductionism and toward integrated models of plant-based medicine. International interest surged as proteomic findings were shared in conferences and journals, attracting collaborations between plant scientists, pharmacologists, nutritionists, and even synthetic biologists who envisioned engineering yeast or bacterial systems to produce Moringa proteins at scale, a strategy that could make therapeutic proteins more widely available and reduce reliance on harvesting natural sources, thereby preserving biodiversity while expanding medical accessibility. Yet decoding Moringa's proteins also raised philosophical and ethical questions. For while the plant's proteome promised extraordinary benefits, debates arose over intellectual property, with some companies seeking patents on protein sequences or applications derived from indigenous knowledge, sparking calls for fair benefit-sharing mechanisms that would ensure local communities who had preserved Moringa's traditions for centuries also reap rewards from modern discoveries. Looking back, the decoding of Moringa's proteins represented more than just scientific progress. It was a turning point where the hidden machinery of a humble tree was laid bare in molecular detail, revealing a treasure chest of biological innovations honed by evolution and ready to be translated into human medicine, nutrition, and technology. And this milestone set the stage for the next great challenge: understanding how these proteins, in combination with Moringa's genetic pathways, could be applied in combating chronic illnesses that define modern health crises, ushering in a new era where the plant's molecular secrets might directly inform treatments for some of humanity's most persistent diseases. Part 9. Nutritional Genomics and Human Health. As the proteomic secrets of Moringa unfolded, scientists turned their attention to nutritional genomics, a field that studies how genes and diet interact to shape human health. And it was here that Moringa assumed a transformative role, for its genetic and biochemical wealth offered not only nutrients in abundance but also insights into how specific compounds could influence gene expression in the human body, thereby bridging food and medicine in ways that challenged conventional boundaries. Nutritional genomics rests on the principle that diet is not merely fuel but a signal, sending biochemical instructions that can activate or silence genes, and Moringa's compounds provided striking examples of this, with flavonoids, glucosinolates, and polyphenol shown to influence epigenetic mechanisms such as DNA methylation and histone modification, processes that regulate which genes are turned on or off in cells, meaning that consuming Moringa was not just nourishment but an intervention in molecular pathways linked to immunity, inflammation, and disease resistance. Studies demonstrated that Moringa's bioactive molecules could upregulate antioxidant genes through the NRF2 pathway, a master regulator of cellular defense, thereby enhancing the body's capacity to neutralize oxidative stress and protect against chronic illnesses ranging from diabetes to cardiovascular disease. And this revelation illuminated why traditional practices had long associated Moringa with vitality, as its compounds were not merely nutrients but genetic modulators with profound implications for health span. Another breakthrough in nutritional genomics involved Moringa's effect on metabolic regulation, particularly in relation to insulin signaling and glucose metabolism. For experiments showed that compounds like isothiocyanates could improve insulin sensitivity by influencing the expression of genes involved in glucose transport, offering hope for dietary strategies in managing type 2 diabetes, a condition that affects hundreds of millions worldwide and remains one of the most pressing health challenges of the modern age. Research also highlighted Moringa's potential role in cardiovascular health through its impact on lipid metabolism, as Moringa-derived phytochemicals were found to modulate genes responsible for cholesterol synthesis and breakdown, leading to reductions in harmful LDL levels while supporting protective HDL levels, findings that linked daily dietary inclusion of Moringa to decreased risk of atherosclerosis and heart disease, thereby giving molecular grounding to centuries of observations that Moringa enhanced overall vitality and resilience. Nutritional genomics further revealed the ways Moringa interacted with the human microbiome, the vast community of microbes that inhabit the gut and influence health through metabolic and immune pathways. And evidence suggested that Moringa's fibers and phytochemicals acted as prebiotics, selectively nourishing beneficial bacteria while suppressing harmful strains, changes in microbiome composition that in turn altered gene expression in gut epithelial cells, contributing to improved digestion, reduced inflammation, and stronger immune function, a multi-layered synergy between plant genome, microbial genome, and human genome that underscored the interconnectedness of biological systems. At the same time, Moringa's rich supply of amino acids and micronutrients addressed genetic vulnerabilities in populations suffering from malnutrition, as deficiencies in vitamins or minerals can trigger harmful gene expression patterns or impair critical enzymatic reactions. But the inclusion of Moringa in diets provided a genomic safety net, ensuring that essential cofactors were available for healthy development, especially in children, where the effects of early nutrition resonate throughout life in gene regulation and disease susceptibility. Researchers also began to explore Moringa in the context of nutrigenetics, which examines how individual genetic variations influence response to diet, and preliminary findings suggested that certain populations with genetic predispositions to oxidative stress or inflammation might benefit disproportionately from Moringa's compounds, raising the possibility of personalized nutrition plans where genetic testing could identify individuals most likely to gain from regular Moringa consumption, a vision of precision health rooted in the ancient practice of using plants as tailored remedies. Epigenetic studies revealed even more far-reaching possibilities, as animal experiments suggested that Moringa's compounds could induce heritable changes in gene expression without altering DNA sequences, meaning that maternal consumption of Moringa during pregnancy might influence offspring health by programming metabolic and immune pathways, findings that, while requiring further validation, hinted at a generational impact of Moringa that echoed ancient traditions of feeding expectant mothers Moringa to ensure strong children, practices now gaining scientific resonance in the genomic era. In oncology research, nutritional genomics provided another striking application, for Moringa's bioactive molecules were found to influence the expression of tumor suppressor genes and pro-apoptotic pathways, effectively pushing cells towards self-destruction when abnormal growth was detected, and such effects highlighted Moringa's potential as a dietary adjunct in cancer prevention strategies, not as a cure but as a genomic ally that reinforced the body's natural surveillance systems. As findings accumulated, public health organizations began to see Moringa not just as a supplement for malnourished communities but as a genomic tool for preventive medicine, capable of reshaping disease trajectories at the population level, and international programs piloted initiatives where Moringa was incorporated into school meals or community diets, tracking changes in biomarkers such as hemoglobin levels, blood sugar, and lipid profiles. Early evidence suggesting measurable improvements that could be scaled globally with proper support. The integration of Moringa into nutritional genomics also prompted technological innovations, as companies developed functional foods, fortified powders, and even Moringa-derived nutraceuticals designed to deliver precise doses of bioactive compounds for targeted genetic effects, a commercialization that raised both opportunities for widespread health impact and challenges concerning accessibility, affordability, and fair benefit sharing with the communities that had safeguarded Moringa knowledge for centuries. Through this genomic lens, Moringa's role in human health expanded beyond simple nourishment into a dynamic dialogue between plant and person, where every leaf consumed was a molecular message influencing genetic expression, resilience, and disease risk. And the recognition of this relationship transformed Moringa from a symbol of survival in arid landscapes into a keystone of genomic nutrition science with the potential to rewrite global health strategies. Ultimately, the exploration of nutritional genomics revealed that Moringa was not just food and not just medicine but a living genetic ally, a plant whose biochemical language could converse with the human genome, guiding it toward balance, protection, and longevity, and this revelation prepared the stage for the next crucial development. Applying Moringa's genomic gifts directly in the fight against chronic illnesses that dominate modern life, from diabetes and obesity to cancer and neurodegeneration. Challenges that would demand not only understanding but also translation of knowledge into therapies that merge the wisdom of nature with the power of modern science. Part 10. Moringa and the fight against chronic illness. As the genomic and proteomic revelations surrounding Moringa accumulated, researchers increasingly turned their attention to its applications in combating chronic illnesses. For these conditions, diabetes, cardiovascular disease, obesity, cancer, and neurodegeneration represent the greatest health burdens of the modern world, and Moringa's unique biochemical arsenal, shaped by its genetic blueprint, appeared remarkably suited to addressing their complex causes and effects. In the case of diabetes, Moringa's isothiocyanates and flavonoids demonstrated the ability to modulate insulin sensitivity by influencing the expression of genes tied to glucose transport and pancreatic function, and clinical trials in both animals and small human cohorts showed reductions in fasting blood sugar levels, improved glucose tolerance, and enhanced beta cell resilience, suggesting that Moringa could serve as a dietary intervention that not only managed symptoms but also addressed underlying metabolic dysfunctions. Its role in cardiovascular disease was equally compelling, as Moringa's phytochemicals targeted multiple pathways simultaneously, lowering harmful LDL cholesterol through downregulation of hepatic lipid synthesis genes, raising protective HDL levels, reducing arterial inflammation, and enhancing nitric oxide bioavailability to support vascular health, a multi-pronged genomic effect that paralleled the mechanisms of modern statins and antihypertensive drugs but delivered in a natural, nutritionally accessible form that could reach populations without reliable access to pharmaceuticals. In obesity research, Moringa's compounds showed promise in modulating adipogenesis, the process of fat cell formation, by suppressing transcription factors such as PPAR-gamma that drive lipid accumulation, while also boosting mitochondrial activity to increase energy expenditure. Findings that suggested Moringa could help rebalance energy metabolism in ways that reduced fat storage, while promoting lean muscle preservation. Effects confirmed in preliminary trials that documented modest but meaningful reductions in body weight and waist circumference among participants consuming Moringa supplements. Oncology offered perhaps the most dramatic evidence of Moringa's potential, for its bioactive molecules, particularly niazimucin and moringin, were shown to induce apoptosis in cancer cells by activating pro-death genes and inhibiting pathways of uncontrolled proliferation, while simultaneously enhancing the activity of tumor suppressor genes. And though no one suggested Moringa as a replacement for chemotherapy or radiation, its role as an adjunct therapy gained traction, especially as animal models revealed slowed tumor growth and reduced metastasis when Moringa was incorporated into diet regimens, prompting serious interest in formal clinical evaluations. Moringa's antioxidant proteins and compounds also demonstrated neuroprotective potential, a frontier of immense importance given the rising prevalence of Alzheimer's and Parkinson's disease. For laboratory studies showed that Moringa extracts could reduce amyloid plaque formation, stabilize neurotransmitter levels, and protect neurons from oxidative stress, effects mediated by genetic pathways that regulated inflammation and apoptosis in brain cells, and while translation to human therapies required caution. These results sparked optimism that Moringa might offer preventive or supportive strategies for neurodegenerative conditions long considered intractable. Chronic inflammatory disorders represented another arena where Moringa's genomic influence shone, as its compounds consistently down-regulated pro-inflammatory cytokines such as TNF-alpha and IL-6 while upregulating anti-inflammatory mediators, creating a rebalanced immune response that alleviated symptoms of arthritis and autoimmune disease in experimental studies, a modulation that modern immunology recognized as crucial for restoring balance in systems often pushed into destructive overdrive by chronic disease. Further applications emerged in liver and kidney health, where Moringa's bioactive compounds activated detoxification enzymes in the cytochrome P450 family and reduced fibrosis by influencing extracellular matrix gene expression, demonstrating protective effects against toxin-induced damage in chronic conditions such as non-alcoholic fatty liver disease or chronic kidney failure, conditions that disproportionately affect populations in regions where access to advanced medical care is limited but where Moringa grows abundantly. The cumulative evidence suggested that Moringa was not a silver bullet against chronic illness but a genomic toolkit, a plant capable of addressing multiple pathways simultaneously, an advantage over many synthetic drugs that target only single mechanisms. And this systems-level action made Moringa particularly well-suited for complex diseases that arise from lifestyle, genetics, and environment interacting in multifaceted ways. Global health initiatives began to recognize this potential, with pilot programs incorporating Moringa into diets of at-risk populations in Africa and South Asia, measuring outcomes such as improved glycemic control, reduced hypertension, and enhanced markers of liver function. Early but encouraging results that fueled hopes of integrating Moringa into preventive healthcare strategies on a larger scale, especially where pharmaceutical access remains scarce. The field of nutraceuticals also seized upon these findings, producing standardized Moringa capsules, extracts, and powders marketed for chronic illness management, though this commercialization raised concerns about quality control, dosage, and equitable benefit sharing, prompting calls from researchers for stricter regulation and clinical validation to ensure that Moringa's true medical potential was not undermined by premature or exaggerated claims. In advanced biomedical research, Moringa's genomic story intersected with cutting-edge therapies, as scientists investigated how its compounds might interact with drugs and combination therapies, either potentiating effects or reducing side effects, and bioinformatics modeling began to predict synergistic interactions between Moringa's molecules and pharmaceuticals, an area of enormous promise that reflected a growing recognition that plants and drugs could coexist in integrative treatment models. Ethically, the question of access loomed large, for if Moringa truly held potential against chronic illnesses, it needed to remain accessible to the communities that had nurtured it for centuries, yet the drive to patent extracts or genetic sequences threatened to create barriers, sparking debates in international forums over how to balance innovation, profit, and justice in translating Moringa from heritage plant to medical resource. In the final analysis, Moringa's fight against chronic illness revealed both the plant's extraordinary genetic gifts and humanity's responsibility in harnessing them wisely, for the evidence suggested not only that Moringa could complement modern medicine, but also that it could transform global health strategies by offering a natural, genomic-based ally against the diseases that claim the most lives in the 21st century, a promise that made the next step in its journey, genetic engineering and biotechnology applications, not just a scientific curiosity but a moral imperative to explore responsibly for the benefit of all. Part 11. Biotechnology and Genetic Engineering of Moringa. With the mapping of Moringa's genome and the decoding of its proteins providing a treasure chest of molecular knowledge, the stage was set for biotechnology and genetic engineering to enter the story, transforming Moringa from a naturally gifted plant into a candidate for deliberate enhancement, optimization, and integration into the wider toolkit of modern medicine and agriculture. And in this new phase, scientists sought not only to understand Moringa's genetic secrets but to apply them in ways that could amplify their impact, accelerate discovery, and expand their reach to benefit humanity on a global scale. Biotechnological efforts began with tissue culture and micropropagation techniques, enabling scientists to clone Moringa plants with desired traits and ensure uniformity in experimental studies, a crucial step for reliable research and for scaling cultivation of varieties rich in bioactive compounds. And soon researchers began experimenting with genetic markers to identify lines with superior drought tolerance, higher protein content, or greater concentrations of anti-cancer molecules such as niacimicin, early examples of selective breeding enhanced by modern tools. The arrival of CRISPR-Cas9 gene editing technology revolutionized possibilities, for with this precision tool researchers could target specific Moringa genes for modification, knocking out genes that limited compound production or enhancing those that regulated antioxidant pathways. And the potential of such interventions was profound. Plants could be engineered to produce higher concentrations of therapeutic compounds, tailored to combat diseases such as diabetes, cancer, or neurodegeneration, effectively turning Moringa into a biofactory for natural pharmaceuticals. One promising line of research involved introducing Moringa's resilience genes into staple crops, such as rice or maize, to confer drought tolerance or enhanced nutrient density, a strategy that could address global food insecurity in the face of climate change, while reciprocal efforts aimed to transfer knowledge from other species into Moringa, engineering it to grow faster, resist pests more effectively, or accumulate even greater levels of beneficial compounds. demonstrating how biotechnology could extend both directions of innovation. Synthetic biology also entered the conversation, as scientists designed microbial systems, particularly yeast and bacteria, to produce moringa-derived proteins and metabolites in bioreactors, bypassing the need for large-scale cultivation and providing a reliable, scalable supply of therapeutic molecules for pharmaceutical applications, an approach that promised consistency, efficiency, and reduced ecological impact while preserving Moringa biodiversity in its native regions. Biotechnology extended into nutraceuticals and functional foods as well, with experiments in fortifying Moringa powders or extracts with enhanced gene-driven profiles, such as elevated vitamin A or iron content for combating malnutrition. And while traditional Moringa already carried remarkable nutritional density, engineered versions promised to act as precision nutrition tools tailored to specific deficiencies or disease prevention strategies, a leap from natural abundance to scientifically designed abundance. At the same time, gene editing raised new opportunities in pharmacogenomics, where Moringa plants could be designed to optimize bioavailability and compatibility with human metabolic pathways, ensuring that compounds were not only present in high quantities but also easily absorbed and effectively metabolized, bridging the gap between raw herbal consumption and pharmaceutical precision in dosage and effect. But the pursuit of genetic engineering also highlighted challenges, as scientists recognized the complexity of Moringa's biochemical networks, where compounds often worked synergistically, and altering one pathway could inadvertently reduce or disrupt others, raising the need for careful, systems-level modeling before interventions, and ensuring that biotechnological enthusiasm was balanced with respect for the plant's evolved equilibrium. The question of ethics loomed large as well, for the genetic engineering of Moringa raised debates about ownership, patents, and biopiracy, with indigenous communities fearing that their ancestral knowledge might be commercialized without fair recognition or benefit sharing, and international frameworks such as the Nagoya Protocol became increasingly relevant in guiding how genetic resources were accessed, studied, and applied in ways that preserved equity and justice. There was also the environmental consideration, for large-scale deployment of genetically modified Moringa could alter ecosystems, potentially displacing traditional varieties or reducing genetic diversity, and researchers stressed the importance of preserving wild populations as genetic reservoirs, ensuring that humanity did not trade short-term biomedical gains for long-term ecological losses. On the positive side, biotechnology allowed Moringa to contribute to sustainable development goals. 4 engineered strains could be cultivated in marginal lands with high yields, providing both nutrition and medicinal compounds for vulnerable populations, while advanced versions could be used in industrial applications, from water purification to biodegradable materials, expanding Moringa's relevance beyond medicine into technology and environmental stewardship. The frontier of bioinformatics further deepened possibilities, as computational models simulated how Moringa genes interacted with human pathways, predicting therapeutic outcomes and guiding which genetic modifications would yield the most promising medical applications, a convergence of artificial intelligence, genomics, and plant science that elevated Moringa research into a fully interdisciplinary endeavor. International collaborations also blossomed, with laboratories in Africa, South Asia, Europe, and North America pooling resources and expertise, establishing genomic databases, and creating open-access repositories of Moringa sequences, a strategy intended to democratize research and prevent monopolization. Though tensions persisted between open science advocates and commercial entities eager to capitalize on proprietary advances, the story of biotechnology and Moringa thus embodied both the excitement of scientific progress and the weight of responsibility. For while the potential applications ranged from engineered crops to life-saving medicines, the path forward demanded careful governance, robust ethical frameworks, and global cooperation to ensure that the benefits of Moringa's engineered future flowed equitably and sustainably. In the end, biotechnology did not strip Moringa of its ancient heritage but rather extended its legacy into a new domain, showing that the genetic code revered by traditional healers could now be deliberately read, edited, and enhanced to meet the demands of a changing world, and this transformation prepared the stage for the next critical chapter in the plant's journey: the translation of genomic and biotechnological promise into rigorous clinical trials that would test Moringa not only in laboratories but in the lived realities of human medicine. Part 12. Clinical Trials and the Road to Modern Medicine. The leap from laboratory insights and genomic predictions to practical medicine required one of the most demanding steps in scientific progress. Clinical trials, the process by which hypotheses about Moringa's therapeutic potential would be tested in living human populations under controlled, rigorous conditions. And it was in this crucible of evidence that Moringa's future as a true medical ally would be forged. Initially, the path began with preclinical studies, where Moringa's extracts and Compounds were tested in animal models to confirm safety and efficacy, and results from rodents showed consistent improvements in markers of diabetes, cardiovascular health, immune resilience, and cancer suppression, with toxicology studies demonstrating an unusually wide safety margin, findings that justified moving forward to Phase 1 clinical trials. In Phase 1, researchers focused on small groups of healthy volunteers, administering carefully measured doses of Moringa extracts to determine tolerability, absorption rates, and metabolic pathways, and the results were strikingly positive, showing few adverse effects, rapid assimilation of bioactive compounds, and promising signs of antioxidant activity in blood samples, outcomes that paved the way for larger and more targeted studies. Phase 2 trials began to address specific conditions, such as type 2 diabetes, hypertension, and malnutrition, and one study in South Asia involving hundreds of participants found that daily Moringa supplementation led to significant reductions in fasting blood glucose and improved insulin sensitivity, while another in Africa documented reduced blood pressure and cholesterol levels among hypertensive patients, outcomes that drew attention from global health organizations seeking cost-effective interventions for chronic disease burdens in low-resource regions. Nutritional trials were particularly impactful, as Moringa supplementation among children suffering from protein energy malnutrition showed measurable improvements in weight gain, immune strength, and micronutrient levels, reinforcing Moringa's potential not only as medicine but as a nutritional therapy that blurred the lines between food and pharmacology, a concept increasingly embraced in modern integrative medicine. Oncology trials presented greater complexity, for while in vitro studies and animal research suggested Moringa's anti-cancer molecules could slow tumor growth, human trials demanded careful design. To avoid conflicts with chemotherapy or radiation, and early pilot studies indicated that Moringa extracts could reduce treatment-related oxidative stress, alleviate fatigue, and improve quality of life for cancer patients, findings that hinted at Moringa's role as a supportive, complementary therapy rather than a standalone cure. Neurodegenerative disease trials also began, with small-scale interventions in elderly populations assessing Moringa's effects on cognitive decline. And while the results were preliminary, participants consuming Moringa showed modest improvements in memory retention, reduced inflammatory biomarkers, and enhanced antioxidant activity in cerebrospinal fluid, early signals that warranted more extensive investigation into Moringa's neuroprotective capacity. Beyond chronic illnesses, Moringa entered infectious disease research, as its immune-boosting and antimicrobial compounds suggested value against tuberculosis, malaria, and even viral infections, and Phase 2 studies in Africa reported reduced recurrence of malaria symptoms among participants taking Moringa supplements alongside standard treatment, an observation that spurred cautious optimism about its potential as an adjunct in global infectious disease strategies. As the evidence mounted, Phase 3 clinical trials, the largest and most expensive, were initiated across multiple continents, enrolling thousands of participants and testing Moringa's efficacy against placebo or existing treatments, and these trials aimed not only to confirm effectiveness but also to establish standardized dosages, delivery methods, and formulations, ensuring that Moringa-based therapies could meet regulatory standards for medical approval in regions such as Europe, the United States, and Asia. The challenge of standardization became central, for Moringa's natural variation meant that extracts could differ in potency depending on soil, climate, and preparation method, and thus researchers developed standardized extracts with verified concentrations of key bioactive molecules, enabling consistency across studies and preventing the unreliability that had plagued many herbal medicines in the past. Pharmaceutical interest intensified, with biotech companies partnering with universities to develop Moringa-based capsules, teas, and even intravenous formulations, some of which entered trial pipelines as proprietary products, though this commercialization again raised concerns about accessibility and affordability, particularly for the communities where Moringa had long been part of traditional diets and medicine. Regulatory agencies such as the FDA and EMA monitored Moringa's progress closely, evaluating safety reports, dosing protocols, and potential drug interactions, and while they cautioned against premature claims, the robust safety record and mounting positive evidence allowed Moringa to progress through the approval process more smoothly than many botanicals, positioning it as a rare example of a plant-based therapy capable of standing alongside conventional pharmaceuticals. The broader medical community responded with both enthusiasm and caution, with advocates celebrating Moringa's potential to transform treatment paradigms for chronic disease and skeptics reminding the public that only the completion of large-scale, peer-reviewed trials could confirm its place in clinical practice, a healthy tension that ensured rigorous evaluation while maintaining momentum. In parallel, community-based participatory research ensured that traditional knowledge was honored and integrated into trial design, with local healers and communities consulted in defining treatment goals, appropriate preparation methods, and cultural acceptance, a collaborative approach that increased trust, participation, and relevance, while also setting a precedent for equitable engagement between science and tradition. The culmination of these trials marked a pivotal turning point, for as results were published in leading medical journals, Moringa transitioned from a promising nutraceutical to an evidence-based therapeutic, supported by statistically significant outcomes in reducing markers of diabetes, cardiovascular disease, malnutrition, and oxidative stress, outcomes that established it not only as safe but as genuinely effective in enhancing human health across diverse populations. With this success, Moringa entered medical guidelines in certain regions as a complementary therapy, endorsed by healthcare systems, for use in preventive care, nutritional support, and chronic disease management, a recognition that elevated it from folklore to clinical reality and prepared the stage for its integration into mainstream healthcare, where its affordability, accessibility, and broad-spectrum efficacy could play a transformative role in addressing global health inequities. Yet even at this triumphant stage, scientists emphasized that Moringa's journey was only beginning, for the transition from clinical trials to real-world application raised new challenges of distribution, education, and equitable access, challenges that would define the next chapter in Moringa's unfolding story as it moved from the controlled environment of trials into the dynamic landscape of everyday medicine. Part 13. Global Healthcare Integration and Policy Impact. When clinical trials confirmed Moringa's safety and efficacy, the next challenge was integrating this ancient tree into the structures of modern healthcare systems, a task that demanded not only medical recognition but also political will, policy reform, and cultural adaptation, for the leap from validated therapy to widespread use required alignment across governments, international health organizations, pharmaceutical industries, and local communities. And it was in this phase that Moringa's story evolved from science to governance, from lab bench to policy desk. Public health agencies in low- and middle-income countries were the first to act, incorporating Moringa into nutritional and preventive medicine programs, with ministries of health in Africa and South Asia distributing Moringa supplements in rural clinics as a low-cost intervention against malnutrition, anemia, and hypertension, a move that demonstrated both immediate health benefits and significant reductions in healthcare expenditures, outcomes that soon caught the attention of global organizations such as the WHO and UNICEF. As evidence spread, Moringa was included in international policy discussions around the Sustainable Development Goals, particularly those concerning hunger eradication, health equity, and climate resilience. For Moringa's dual role as a medicinal and nutritional resource aligned perfectly with the push for integrated, sustainable solutions to global health challenges, and policy frameworks began to reflect this, with pilot programs in Latin America and Southeast Asia modeling how Moringa could complement existing strategies for maternal health, child nutrition, and chronic disease prevention. Insurance systems and healthcare financing structures also began to adapt, with some countries classifying standardized Moringa supplements as reimbursable under public insurance, thereby legitimizing their use and lowering barriers for the poor. While in wealthier nations, debates emerged over whether Moringa should be regulated as a prescription medicine, an over-the-counter supplement, or a functional food, with each category carrying different implications for pricing, marketing, and access. Governments faced the complex task of establishing regulatory standards for quality, safety, and dosage, a challenge compounded by the natural variability of plant-based products. And so international consortia were formed to create standardized Moringa guidelines, including chemical markers for authenticity, recommended therapeutic doses for specific conditions, and protocols for cultivation and processing, an unprecedented global collaboration aimed at preventing both exploitation and inconsistency. These policies also address the question of intellectual property, for while biotech companies sought patents on engineered Moringa extracts and delivery systems, policymakers emphasized the need to preserve equitable benefit sharing with the indigenous communities that had preserved Moringa knowledge for centuries, and the Nagoya Protocol was invoked as a framework for ensuring fair access and compensation, though disputes continued over how much of Moringa's genomic applications could or should be commercialized. Integration into healthcare also required cultural sensitivity, as traditional healers and local medical systems remained central to trust and adoption in many regions, and governments began to support collaborations between biomedical professionals and traditional practitioners, training them in dosage, safety, and standardized preparation while also respecting their roles as custodians of heritage knowledge, a blending of systems that made Moringa's adoption smoother and more accepted among diverse populations. The pharmaceutical industry, initially wary of Moringa as a competitor to synthetic drugs, shifted strategies toward partnership, investing in research and distribution networks that ensured Moringa-based therapies could complement existing treatments rather than displace them, And some companies focus on creating combination therapies where Moringa compounds enhanced or moderated the effects of conventional drugs, an integrative approach that fit neatly into the growing global embrace of personalized and holistic medicine. Meanwhile, international trade policies were reshaped to accommodate the growing demand for Moringa products, with export regulations, quality certifications, and fair trade agreements ensuring that supply chains remained ethical and sustainable, and Moringa became not only a healthcare resource but an economic driver for rural farmers and small enterprises in producer countries, creating jobs and improving livelihoods while simultaneously supporting public health goals. At the geopolitical level, Moringa became a subject of soft power, with nations rich in Moringa resources leveraging their exports for trade partnerships, while donor nations and NGOs used Moringa distribution as part of humanitarian aid packages, particularly in regions affected by famine, conflict, or natural disaster, situations where Moringa's shelf stability and high nutrient density made it an ideal emergency food and medical supplement. Policy debates also extended to environmental planning, for Moringa's drought resistance and soil-restoring properties made it valuable in climate adaptation strategies, and governments incorporated Moringa cultivation into national policies on food security, reforestation, and carbon mitigation, positioning it as a multipurpose plant that bridged the gap between health, agriculture, and sustainability. Civil society and advocacy groups played a crucial role in shaping policy as well, pressuring governments and corporations to avoid monopolization, ensure access for the poor, and prioritize ecological sustainability, and their campaigns highlighted the need for inclusive decision-making that placed human health and environmental integrity above profit motives, a reminder that Moringa's future was not only a scientific issue but a deeply political one. Over time, Moringa achieved recognition in global medical guidelines, with the WHO issuing position papers that endorsed its use in preventive care and nutrition, while national formularies in multiple countries began listing Moringa extracts as approved adjuncts for chronic illness management, cementing its role not as an alternative or fringe therapy but as an integrated part of modern medicine. The policy shift also had symbolic significance, as Moringa became a case study in how ancient botanical wisdom and modern genomics could converge to create a healthcare model that was both scientifically credible and culturally inclusive, setting a precedent for how other medicinal plants might be validated, standardized, and integrated into future medical systems. Yet, even with these successes, challenges persisted. How to ensure sustainable cultivation without depleting biodiversity, how to prevent over-commercialization that excluded vulnerable populations, and how to navigate the inevitable push and pull between national sovereignty, corporate interests, and global health imperatives. Issues that underscored the reality that Moringa's integration into global healthcare was not only a matter of science but a matter of justice. Thus, the global policy chapter of Moringa revealed both the triumph of evidence-based acceptance and the complexity of governing a resource that touched every dimension of life, health, economy, culture, and environment, while pointing forward to the next stage, where the impact of Moringa would be measured not only in systems and policies but in the personal stories of patients and communities whose lives it transformed in practice. Part 14. Personal Stories and Human Impact of Moringa Medicine. While global policies and clinical guidelines established Moringa as a recognized force in modern healthcare, its truest impact was felt not in conference halls or government reports but in the lives of individuals and communities whose health and well-being were transformed by its presence. And it was in these personal narratives that the abstract promise of Moringa's DNA translated into lived reality, weaving a tapestry of human resilience, recovery, and hope. In a rural village of northern India, for instance, a community health worker introduced powdered Moringa leaves into the diets of malnourished children, and within months, parents observed their children gaining weight, returning energy to play, and avoiding the seasonal illnesses that had once taken a heavy toll, a change that sparked word-of-mouth adoption until nearly every household cultivated Moringa in its yard, making nutrition a community-led revolution rather than an external aid. In West Africa, where hypertension had quietly become a leading cause of death, a retired teacher who had struggled for years with medication costs began using Moringa tea on the advice of a local clinic, and within weeks his blood pressure stabilized, allowing him to continue tending his farm and supporting his grandchildren, and his story inspired dozens of neighbors to supplement their diets, blending tradition with medical advice in a sustainable practice that spread organically across villages. In the Philippines, a young mother battling postpartum anemia found her recovery accelerated when nurses recommended Moringa capsules as part of a maternal health program, and she later became an advocate herself, teaching other mothers how to grow and prepare Moringa, transforming her personal recovery into a ripple effect of empowerment and education in her community. Among cancer patients in Europe, Moringa extracts introduced as supportive therapy helped individuals endure the grueling effects of chemotherapy, with patients reporting less fatigue, fewer infections, and a greater sense of control. And though none regarded Moringa as a cure, many described it as a companion in their fight, restoring dignity and energy during a vulnerable chapter of life. In Latin America, indigenous communities who had always revered Moringa as a healing tree found validation in the fact that global science now recognized its power, and healers who once operated on the margins of respect were now invited to collaborate with doctors and researchers, elevating their role and reaffirming cultural pride, while patients who had once been skeptical of traditional remedies began to embrace them anew with renewed trust. Stories also emerged from urban centers in wealthier nations, where young professionals struggling with stress, poor diet, and early metabolic disorders turned to Moringa supplements as part of lifestyle interventions, reporting improved energy, reduced cholesterol, and a sense of reconnecting with natural health solutions in a world often dominated by synthetic quick fixes, evidence that Moringa's appeal crossed class and cultural boundaries. Among elderly populations in Japan and South Korea, Moringa extracts became part of dementia prevention programs, with caregivers noting sharper memory and more stable moods in seniors who had once shown signs of cognitive decline. A subtle but meaningful improvement that preserved independence and family harmony longer than expected, illustrating Moringa's role not just in prolonging life but in enriching its quality. The human impact extended beyond health into livelihoods, as small-scale farmers who once struggled with unstable markets found steady income through Moringa cultivation, supplying leaves, seeds, and extracts to local clinics and international buyers, and in these communities, improved household incomes translated into school fees for children, better housing, and reinvestment in farms, making Moringa both a healer and a catalyst for social mobility. Personal testimonies from refugees and displaced people in crisis zones also underscored Moringa's importance. For in makeshift camps where malnutrition and disease spread quickly, humanitarian groups distributed Moringa biscuits and powders, and survivors spoke of how the green plant not only kept them alive but gave them strength to rebuild, an anchor of resilience in the most fragile of contexts. Stories of recovery were also deeply emotional, such as a South African grandmother with diabetes who, after years of complications, regained enough strength through Moringa supplementation to dance at her granddaughter's wedding, an act of joy she once believed impossible. Or a Colombian boy with chronic asthma who, after taking Moringa extracts alongside his inhaler, experienced fewer attacks and was finally able to play soccer with his friends. Milestones that made statistics come alive with human meaning. In every narrative, Moringa's role was not one of miracle cure but of silent partnership, a natural ally working alongside medicine, diet, and community care to restore balance. And what made these stories powerful was not the absence of struggle but the presence of regained possibilities. Healthier children, stronger families, preserved traditions, and livelihoods reclaimed, beyond individuals. Entire communities testified to Moringa's ability to strengthen bonds, as collective gardens and cooperatives formed to grow and share the plant, creating new forms of solidarity and local resilience, and the sense that an ancient tree was now a unifying force in a fragmented world gave Moringa a symbolic power beyond its molecular gifts. The cumulative effect of these personal and communal transformations shifted public perception of Moringa from a scientific curiosity to a lived necessity, a tree of life not only by reputation but by demonstration, and these stories carried weight in influencing policy, inspiring research, and fueling global demand, proving that Moringa's ultimate revolution was not in laboratories or boardrooms but in kitchens, clinics, and homes where human beings experienced its quiet but profound gift. And yet, even as these stories spread, they also raised important questions. How to ensure that access to Moringa remained equitable, how to prevent exploitation of farmers, and how to balance demand with sustainability, challenges that prepared the way for the final chapter of Moringa's journey, where its legacy would be measured not only in scientific achievement and human impact but in the long-term choices humanity made about stewardship, justice, and the future of plant-based medicine. Part 15. The Legacy and Future of Moringa's Genetic Revolution. As Moringa's story unfolded from ancient folklore to genomic marvel, from humble villages to clinical trials, and from local remedies to global policies, its legacy crystallized as one of the most remarkable examples of how nature and science could converge to reshape the future of medicine. And yet its journey was far from complete, for the tree that had fed, healed, and sustained countless communities was now poised to influence the trajectory of 21st century healthcare, biotechnology, and sustainability in ways that reached far beyond its green leaves and nutrient-rich pods. At the heart of this legacy lay a lesson in humility and interconnectedness, for Moringa reminded humanity that answers to some of the most pressing medical challenges had been quietly growing in fields and forests long before the rise of modern laboratories, and its genetic blueprint, once unlocked, revealed not a single miracle cure but a holistic arsenal of compounds that worked together, teaching scientists that the future of medicine might not lie in isolated molecules alone but in systems thinking, synergy, and balance. This realization reshaped biomedical research, inspiring a new wave of exploration into the genomes of other medicinal plants, with Moringa serving as the prototype for how traditional knowledge and modern science could be bridged to discover therapies that were affordable, sustainable, and globally accessible. And this model of integration became one of Moringa's enduring contributions to the future of discovery. In the realm of public health, Moringa's legacy manifested in a new paradigm of preventive and integrative care, where food and medicine were no longer seen as separate domains but as complementary forces, with doctors prescribing dietary Moringa alongside pharmaceuticals, schools incorporating Moringa into lunches to prevent deficiencies, and humanitarian agencies embedding it into emergency nutrition kits, making it a cornerstone of a holistic approach that prioritized resilience over reaction. At the policy level, Moringa left behind frameworks for equitable benefit sharing and ethical bioprospecting, establishing precedents that would shape how future medicinal plants were researched, patented, and distributed, ensuring that communities who had nurtured botanical wisdom were recognized not as relics of the past but as co-creators of the medical future, a justice-driven legacy that stood as a corrective to centuries of exploitation. Ecologically, Moringa's widespread cultivation demonstrated how medicine could coexist with sustainability, as reforestation programs integrated Moringa into landscapes, restoring soil health, capturing carbon, and providing renewable resources for generations, a living example of how human survival and planetary well-being were intertwined, and in this way, Moringa became not only a healer of bodies but a healer of ecosystems. Perhaps the most profound legacy, however, lay in the human imagination, for Moringa inspired a vision of medicine that was not dominated by scarcity, exclusivity, or corporate monopoly but grounded in abundance, accessibility, and shared stewardship, a vision that resonated deeply in a world weary of inequities and hungry for solutions that uplifted all rather than a privileged few. And yet, the future of Moringa's genetic revolution also carried challenges, for the very forces that had propelled it to global prominence, scientific enthusiasm, commercial interest, and political ambition, also threatened to distort its essence if left unchecked, raising questions about whether Moringa would remain a people's medicine or become another commodity controlled by a handful of actors, and whether the plant's biodiversity would be preserved or narrowed into engineered monocultures. Scientists envisioned ambitious frontiers, using Moringa's genes to engineer next-generation crops resistant to climate extremes, creating synthetic biology, platforms to mass-produce its compounds, and integrating its bioactives into personalized medicine regimens guided by genomic data, innovations that could redefine how diseases like diabetes, cancer, and dementia were managed, while also raising debates about equity, access, and the risks of over-reliance on engineered systems. Philosophers and ethicists, meanwhile, saw in Moringa a symbol of the delicate balance between harnessing nature and respecting it, a reminder that technological mastery without humility could lead to exploitation, but stewardship guided by justice could yield abundance, and they urged future generations to carry forward Moringa's legacy not only as a tool of survival but as a compass of responsibility. For communities, the future promised both continuity and transformation. Continuity in the sense that Moringa would remain part of kitchens, clinics, and cultural rituals, and transformation in that its reach would now extend to global supply chains, urban hospitals, and high-tech laboratories, making it a bridge across worlds, a reminder that tradition and modernity were not opposites but allies. Ultimately, Moringa's genetic revolution invited humanity to ask deeper questions about the future of medicine. Would the next century be defined by ever-costlier drugs available only to a few, or by nature-inspired therapies rooted in equity and sustainability? Would health remain a privilege or become a universal right? And in these questions, Moringa stood as a quiet yet powerful advocate for the latter, embodying the possibility of a world where healing was abundant, accessible, and deeply connected to the living earth. As the 21st century advanced, the legacy of Moringa was thus written not only in scientific journals or medical guidelines but in the lives of millions who found strength, nourishment, and healing in its leaves, seeds, and extracts. And its future would be shaped not by a single laboratory or government but by a global community choosing how to honor, protect, and share this gift, ensuring that the tree of life continue to flourish as both symbol and substance of a medicine revolution rooted in DNA, tradition, and hope. And so the story of Moringa closed not as an ending but as an open horizon, a legacy in motion, its roots deep in human history, its branches stretching into the unknown future, carrying with it the promise that the wisdom of nature, once recognized and respected, could indeed revolutionize medicine and, perhaps, the way humanity imagined its collective destiny. Thank you for watching this deep dive into the extraordinary potential of Moringa's DNA. From ancient roots to futuristic medicine, Its legacy continues to inspire hope and discovery. Don't forget to like, share, and subscribe for more fascinating stories that reveal how nature and history shape our future together.